Hello, welcome to part 2 of 3-part lecture series. In last lecture, I had talked about quantum effects. In part 2 today, I'm going to talk about origin of life. And in part 3, I will talk about consciousness, which will deal with brain activities. My name is Sangam Banerjee and my details are given below. So let us start with origin of life. This is part 2 of 3. So let us start with Big Bang. So we know the universe was created some 14 billion years ago. We are here 14 billion years and the Big Bang started. And in first second you had elementary particles which was created. And then due to interactions, atoms and molecules were formed. And after 300,000 years, you get this particle dust cloud. And after 1 billion years, the universe cooled and started forming galaxies. And now we are here after 14 billion years. So Earth was formed some 4.5 billion years. This is a red hot Earth. It cooled down. And then after cooling down, molecules were synthesized, by Earth. Here is a prebiotic chemistry. And this molecule, they self-assembled and formed some pre-RNA world. And then this RNA started folding, and folded RNA has some functional form, which we will discuss. And then from RNA in 3.6 billion years ago, it is believed that first DNA and protein life started. Then around 3.6 billion years, you have this LUCA, that is last universal common ancestor, and then diversification of life started. We are going to discuss. We will mainly discuss what happened before this diversification, and then maybe we will see how this living organism started evolving. So as I said, you had this plankton. So oxygen was created due to this tiny plant and animal. They were wandering and drifting in the ocean, and this plant, this phytoplankton means is a plant, produce 40 to 60% of oxygen we breathe, and together with zooplankton create a food web that supports all other marine life. So to recapitulate, 14 billion ago you had a Big Bang, then Earth was formed around 4.8 billion, and these are the timeline. 2 billion years you had eukaryote, and Homo sapien man appeared some 50,000 years ago. So you just freeze and see this timeline. And how do you know this timeline? That is from geological survey. They do it. They go layer by layer on the earth crust, and these are called the eras of geological time scale. So the more deeper you go, the more backward in time you go, and this is how they find fossils, and thus they record the history of evolution. And here I have shown pictorially, these are the fossils, the same thing here, that how it evolves, and this is the evolutionary how it evolved in a GIF format. So it is believed a complex molecule required for life form started from primordial soup, this prebiotic chemistry. Some feel that it has come from some earlier molecules, protein or some amino acid might have come from outer space, but mostly many group, they feel that no, it originated in our earth only, in a hydrothermal vent, which I will show you. And then you can see that once you have this, how reactions are taking place, and you have different organic molecules like methane, carbon dioxide, hydrogen cyanide, and how these are made, we will see. So origin of life timeline, again I said, you had a Big Bang, then you had no life here, some chemical elements were synthesized. This is given in terms of billion years up to then organic compound was synthesized in space, that people have found evidence from meteorites. And then Earth was formed, and then as I said, last universal common ancestor was formed. Earlier fossil you can see, then photosynthesis, atmospheric oxygen was generated, then sexual reproduction, and then you had a biological Big Bang, which I showed, that micellar life and other kind of life. Earlier, which I showed, was a molecular Big Bang, then you had a biological Big Bang. So as I said earlier, that this is a hydrothermal vent in deep sea. They are very high temperature volcanic gases. They get erupted, and there are a lot of atoms and molecules, and they self-assemble by, these all require, we will see, we saw in our last lecture how quantum mechanics helps. One conformation to another conformation molecule can go by tunneling. So in this hydrothermal vent, these are the clay, and the clay, these microcrystallites were there in the clay. This clay is composed of crystallites, and of course they acted like a substrate, like a dye, and then these atoms and all, they got assembled here, and they act like a catalyst also. They can form a very complex, this is nucleotide polymerization can happen. And so how do you know this thing happened. There are some geological evidence. You have this hydrothermal pool in Atlantic Ocean, and in certain part of the world, you can, some geological evidences. Scientists have found some using microscopic technique, such kind of molecules. Now we discuss chemistry to RNA-based cellular structure. So there are two hypotheses: RNA hypothesis and lipid world hypothesis. These are the two competing theory going around. So anyway, you have a primordial soup which consists of sugar, complex sugar. Then you have lipid membrane. We'll discuss little bit what lipid membrane is. Then you had amino acid which was synthesized in this primordial soup. And then of course, as I said, that polymerization of this amino acid had taken place, and polymerization of nucleotide gave rise to RNA and DNA. And then of course you have this lipid membrane here, and these parts were incorporated. We will see this. So what is this lipid membrane? We know we daily use soap. So this soap has some hydrophilic part, means it likes water, and it has a tail of CH, carbon and hydrogen chain, and it doesn't like, it's like an oil. Okay, they want to be together, and so this is the hydrophilic part and this is the hydrophobic part. Okay, so when you have an oil or non-polar dirt on cloth or on your skin, this hydrophobic, this chain gets attached to this oil, and this part is hydrophilic, and then they will keep attaching, and then it will remove the dirt. Okay, so this is how your amphiphile, the soap, works, which we all know. And this amphiphile can form various shapes. This is called micelle, and if it forms a double layer, it's called liposome or bilayer sheaths. Okay, so this kind of structure were also formed in the primordial soup. And as I said, primordial molecular synthesis is called abiogenesis, that depending on carbon oxygen ratio, when there was more carbon and less oxygen, and with the help of sun rays, especially the UV, there was not much atmosphere earlier, so a lot of molecule synthesis happen. And there are some molecule, like when your carbon is more than oxygen, so carbon rich molecules were generated, and when carbon oxygen were equal, like carbon monoxide. Okay, so this kind of stuffs were synthesized using the sun rays and atmosphere. And also some people believe some of the molecules might have come from comet, and in earlier they were lightnings. So with the help of this lightning, then the sun rays, a lot of molecules have synthesized. This was done synthetically by Miller in experiment also. You can do these things in lab. So here I have shown that when you have, say, hydrogen cyanide here, HCN, this in the presence of water, such kind of molecule is called EAMN. The full form is here. And with the help of UV, this molecule can transform to this, and some ratios, some goes to again HCN, and the reaction can continue and can produce nucleotides like adenine and guanine. So from simple molecule, one could get this complex molecule due to chemical reaction. Then emergence of behavioral chemistry, this is autocatalytic Formose reaction. So here what happens is that you can keep on adding carbon. So it is interesting that the carbon is the most important constituent for life. So carbon to carbon condensation started taking place, and this is autocatalytic reaction. This is called the Formose reaction. So it kept on adding carbon, and various molecules were synthesized. And here also you see, if one synthesizes formamide, then you can get this kind of amino acid. You can show, actually, you have to believe that people can synthesize in the lab this kind of from this molecule. Then you can have nucleobase, carboxylic acid. Then this is your nucleosides. Okay, and then you have the sugar molecule. These are all carbon here. It has not been written. And then you have amino sugar also. Okay, so whenever there is a junction, it is mostly carbon. So they don't write so much carbon is this, so they avoid writing carbon all the time. So we saw in primordial soup, you have this part of this nucleosides here. This contain nitrogen, so they are called nitrogenous base. And when they connected here, this total is a nucleotide. And this nucleotide with chemical reaction can polymerize and form an RNA chain. So there are three types of RNA. This one is called the messenger RNA, and then you have a ribosome. Ribosome also contains portion of RNA and protein. The main function of a ribosome is to read this messenger RNA, and with the help of transfer RNA, which carries an amino acid, it will, here I have shown the animation that this transfer RNA comes, the ribosome read it, and then the ribosome synthesize this protein, which is or amino acid chain, which further folds and form a functional protein. So this is how protein synthesis take place inside a cell. Now from chemistry to biology. So the prebiotic world is a dead state. That is, you just have precursor, then you make nucleotides by abiotic chemistry, then random polymerization to make RNA. And very rarely it can form ribosome too. And biology happens with the RNA world. This is an autocatalytic living state. So you have a precursor, then you have some biosynthesis, you make nucleotides, then you have RNA replication, then you make ribosome. Now here you see these all act like a autocatalysis. This fellow will help to make ribosome. This cycle, nucleotide synthase, will make nucleotide. So these are the cycle, that is, these RNA polymerases, they polymerize the nucleotides and make ribosome. Or this ribosome can become nucleotide synthase, and from here you can make nucleotides. And this ribosome can also help in synthesizing the precursor. So metabolic ribosome reduce reliance on the precursor. So this ribosome, they don't really depend on the precursor. Ribosome itself can make a precursor. So metabolic ribosome reduce reliance on precursors. Now what you see here is synthesis of informational RNA, then it folds, become functional RNA like we saw it in ribosome. Then you produce functional protein. Then with the help of functional protein you can get informational DNA. Here I have shown here that you have informational RNA, it folds, functional RNA you get. Then you have from functional RNA you can synthesize protein to get functional protein. And similarly this is the modern molecular biology, where from RNA you have functional RNA, then you have functional protein, and then this functional protein can synthesize DNA. This is the information DNA. And then once you have a DNA, from DNA to DNA you can get. So this is how it works. So to recapitulate, evolution from chemical soup to life, you had some prebiotic chemistry, then you had some biomolecule, then self-assemble here, this lipid bilayer has formed, vesicles, and some this RNA and other stuffs got integrated inside the cell, and then cell started dividing, and then you have present day unicellular organisms. So here I show a vesicle containing some constituents. It interacts with the cell membrane and ejects out these constituent molecules. So this is how sometimes the synapses, when we discuss, it works like this. And during the formation of this, vesicles or cell membrane, many other functional parts got incorporated on the cell membrane. So here I show that this prebiotic possible, that there are this self-assembly is taking place and it forms vesicles. So please freeze it and see the different constituent of this cell, this protocell. And here I say that enzymes are selected only if they fit into protometabolism. So they are selected only if it helps them, otherwise it is discarded in the next generation. So to recapitulate again, I say this is the RNA world hypothesis, that is, it has information storage, inheritance, selection and propagation. So this is the nucleotides are here, RNA, and then RNA replication takes place. So here and then RNA catalyzed protein synthesis. So here you see this is a transfer RNA which carries a amino acid and you form a protein. This is your say ribosome, which I have discussed in earlier slides. Then RNA encodes both DNA and protein. So from RNA you can get DNA and from RNA you can get protein. Then of course it get encapsulated. Cell membrane is formed and you have different constituent, protein, RNA, DNA. And this is that LUCA which I said, last universal common ancestor. And once this is a protocell here, this is, and from this point on we diversified. So this forms three branches. Okay, eukaryotic, then here is eukaryote, then the bacterial branch, and then archaea branch. Okay, and of course from eukaryote you got your plant. So eukaryote means each cell has a nucleus and nucleus contains DNA. Here DNA is scattered across the cell, and this archaea is also very similar to bacteria, only the difference is their cell wall. So one differentiate archaea with bacteria from the cell wall, which maybe we will discuss. So I discussed earlier that every cell wall you have some functional molecule. So this is your ATP synthase, and these are some electron transport channel. This is the cytoplasm of the cell. So they can sit in this cell wall here. So these are your thylakoid in chloroplasts. And this is mitochondria, which is known as a powerhouse. So in the last lecture also we discussed about ATP cycle, where ATP becomes ADP plus phosphate. That is triphosphate, one of the phosphate goes, so it becomes diphosphate. And the energy release is used for doing work, for moving some parts inside the cell. And excess energy goes to the thermal disordered energy. And ADP, with the help of energy, that is when you break sugar and all, energy is released, it forms ATP. Okay, in the this in ATP synthesis is required to make ADP to ATP. Actually, this ATP synthesis so got a Nobel Prize in 1997, but still the structure, people are still doing it and making it much better and better structure. So this is the updated structure in 2016, and maybe in future this structure may further get updated. So here it is adenine type triphosphate. So there are three phosphate. This is the ribose and this is the adenine base. So this is the full structure of ATP. ATP is an important molecule in metabolism because it is the only chemical in the body that can be directly used as energy. Because when you break it, the energy is released and you can do useful work here. This ATP cycle. Now here you see how the ATP works. So you have an ATP. So it binds and it forms ADP and a phosphate, inorganic phosphate. And this fellow, once it release energy, this fellow moves. So that is what I was saying. You see here also you have this actin here, the protein. These are in this kind of cell like amoeba, single cell actins are there, and this is also a protein myosin, and there are some kinase here which keeps moving, and this reaction is always going on. That ATP comes bind here, becomes ADP plus inorganic phosphate, and then again your ATP comes from your ATP synthesis. So here are some animation where I am shown how the ATP comes, releases energy, forms ADP and phosphate. Okay, so here you see a single cell actin. This cell has some receptor. Once they get some chemical signals, the actin starts moving there, so the cell membrane also move. And here you see how cell division is taking place. This is some cell with microtubule. I will come to this also in next slide. So you have two school of thoughts. This is RNA world and a lipid world, which I have discussed in detail. And in a lipid world, they say that everything is mixed together and then they self-organize inside the lipid membrane and then you form the protocell. So in lipid world, it says that self-replication depends on other cellular components and catalytic interaction increases molecular diversity, which happens inside. And protocell grows to twice its original size and then will be ready to split. Okay, so depending on some critical size, this protocell divides. So these are the only things which is happening before the LUCA stage, that is last universal common ancestor state. And here we show the lipid structure of bacteria and eukaryote, which is slightly different from archaea. This has some side chains are there, and these are the phospholipid bilayer for archaea and phospholipid bilayer for bacteria. So the cytoskeleton is the most important organelles. Without it in place, all the other organelles inside the cell would be randomly floating. So this cytoskeleton, like this actin, microtubule, they are the skeleton of the cell. Okay, otherwise all the organelles, they will keep randomly floating. We will see also how this act as a pathway, these microtubules. The virus take this as a highway, they have been transported to nucleus. Otherwise, in the cell, there's so many, you see here so much of proteins are there. It is so jam-packed, and how they go from one place to another place is really mind-boggling. And this is here I show you how the cell division take place. Okay, you have this microtubule, there are centrioles, then they attach to the center portion, centrosome, and then they pull it, and once they pull it, the cell divides. So these are the one which I said, this is the part of these cells. These are centrioles. Then you have microtubules, then these are the chromosomes, and these are centromere. So this microtubule comes and attaches this centromere and they pull it. So how do the cell makes the living by cooperation? So they have some cooperation. That cell, there are some antigen bacteria, antigenic, then methogenic bacteria are also there. These are the bacteria. So they take carbon dioxide, gives acetate. Okay, this fellow takes acetate and gives out methane. So they survive by cooperative, they are cooperating with each other, producing chemicals so that the other survives. So here you see acetogenic bacteria. Okay, they gives acetate here. This fellow takes it. It forms methane. And here also, these are the reaction. So this is a kind of a network where each bacteria, cooperates and provides required chemicals for other bacteria to survive. So here I have shown here, taken it from this lab. This is a synthetic protocells, and you see the cell with time is dividing. And here I have shown what is the role of actin inside a cell. So these actins are there inside the cell, and they are used for their locomotions. Their movement is determined by this actin, and this is how the actin is synthesized. So these are a linear chain of proteins, and here I have shown here that this how eukaryote came into existence is something like one bacteria going inside another bacteria, and cooperatively they are living together. So here I have shown this prokaryotic where DNAs are scattered, and this is a eukaryotic like animal and plant cell where new DNA is inside the nucleus. So here I show that how some viruses has helped changing our DNA by inserting into the cell and integrating with the DNA inside the nucleus. And here you see this HIV virus I have shown, which is also very dangerous. You just simply cannot get rid of the virus because they take their genetic material and it get integrated in the DNA. Once the viral DNA is integrated, it will keep producing viruses. And nature found reshuffling of genetic material is important for evolution. So this is, we have shown here a bacteria. There are three mode of genetic transfer: transduction, conjugation, and transformation. So these are shown here. Like this is transduction, means this virus attacks and then makes a genetic, by mutation it have a new genetic material and again infect the other bacteria. So this is how the shuffling of genetic materials are taken, or this is by conjugation. The bacteria fuses with other bacteria and transfer the genetic material. This is just for completeness sake. Plant cell have a definite cell wall. Animal cell doesn't have a definite. And this is one single cellular organism called euglena. It has a nucleus inside in the center and then different parts. So please freeze it and see this different organelles and parts of the cell. So to recapitulate, we started with very basic molecules, and of course with the help of UV and temperature and so on and so forth, you could get all this complex fatty acid, glycerol, nucleobases, and with these nucleotides you could make RNA, and here with this kind of fatty acid and sugar you could make phospholipids for making cell membrane, and this goes inside, incorporates inside the cell DNA, RNA, and then there are some protein which has been synthesized with the help of RNA, and protein has lot of functional forms and this protein is synthesized and is used for making many structures inside the cell such as actin, microtubule. And then this is how, once you have these things, it is incorporated inside the cell membrane, and this is called the Luca, the earliest life, and then it diversified from here. Okay, bacteria, archaea, and eukaryote. We plant and animals are in this branch. And this is what I had mentioned, how virus enters and use microtubule, and as a highway, it is this fellow is kinase is carrying it, and these constituents are multiplied, and again they have been carried here, and then a vesicle is formed and a new virus is formed. Okay, so just freeze it and see. This is very easy to understand. And here I have shown you, this is a microtubule. So there are many microtubule associated protein, and they have various functions. Like some kinase, they transport vesicles. As I said, even can transport the virus. So for transportation, this is required, and this microtubule is used as a highway. Okay, and this is, it has some plus end and a minus end, and it grows in this direction here. So here I have shown how certain unicellular organism moves with the help of the acting. They migrate and pushes the cell wall, and the unicellular organism can move. And there are receptors, chemical receptors are there in the direction it sends. The actin helps the membrane to move in that direction. So here also some animation I have shown how this is maybe amoeba, and these are different unicellular. And this is a sperm cell. This also has a microtubule here, and there is a mitochondria which provides energy for this microtubule to move. And this is your cell inside. As I said, that it forms like a skeleton. So you have this microfilament here, and there are fibrous subunits. These are all made of protein, and there are microtubules, so they hold the cell intact, so that these organelles, they don't float around. Wherever they are stuck. Okay, otherwise this will keep floating, which is not required. So here I show that there's a directional sensing. There's a receptor chemical. So if chemical comes, this receptor will sense, and it will trigger this actin formation. The actin will start polymerizing here. The actin filament will grow and will pushes the cell wall. So this is how unicellular organism moves, by with the help of this actin will get elongated, will push the cell wall. This is life is imperative. That is, there is always a drive for survival. You see here WBC, and they also, with the help of actin and microtubule, they move, and it is hunting a bacteria here. And it interesting to see that it follows the bacteria because it senses the chemical released by the bacteria. And this organism will sense the chemical gradient and will move towards high density of this chemical. And this is here again I have shown that how actin and microtubules, there are actins here, and microtubule pushes the cell wall and helps the organism to go towards the food. So here I am showing WBC how they go and clear of the bacteria colony, and here there is a macrophage, also how they catches the bacteria and engulfs it. Okay, this bacteria, they release some cytokines, some chemical signals, and they know from where it is coming. And the actin and microtubules, they pushes the cell wall and they grab this bacteria. So here also I show some immune dendritic cell engulfing bacteria. These red ones are the bacteria. One more interesting thing which I found is this bacteria, when it is inside the cell, it uses the cell protein and forms an actin filament. And once it forms an actin filament and it is forming this side, so this actin filament is push behind, so there is a this side push, and due to opposite reaction the bacteria moves forward. So as it keeps producing this comet-like tail. If you see here is a comet-like tail, it will keep producing, and this bacteria will keep moving. Here I have shown these bacteria are moving inside a cell. Okay, here this is a flow, and here I show some electron microscopic picture of microtubules and actin filament inside the cell, and how they are branched, and how this help in pushing the cell membrane. So here I show some animation of this bacteria, same which we showed, that with the help of this actin, they move. Not only move, they can penetrate, they can move from one cell to another cell, and they, while after entering, they divide, multiply, so they can create havoc, and they use your protein from the cell for its locomotion. So here we see a very interesting phenomena. They synthesize a very smart material, this cell membrane, and you take a portion of the cell membrane and you stick it to a polystyrene ball and put it inside a proper nutrient containing tubulin and such protein. And this polystyrene ball coated with this bacteria, just one portion of the bacteria, they also behave. So that means this material itself has a property to create comet-like structure. So here a polystyrene ball which is not coated, and here it is coated, and you see they form a comet. So this is published, and just Google it with this reference, you will find this paper. Now microtubule actin and filamase based cell motility, which we have discussed. So here just I want to show here that this, suppose you have a neuron which also has microtubule, and it is connected to other microtubule, and they can send some information for how the microtubule has to behave. And so here it is interesting to see that from one microtubule to another microtubule, signal can be passed through some filament which keeps the microtubule stable. And here I show the plasticity of neuron. So here also the microtubules helps, like what we saw for single cell organism. So neuron is also unicellular. Here it seems like it is trying to search for another neuron to get connected. So these are all in petris, and this is the time scale at which this neurons grow. Okay, they grow in few seconds, less than a minute. And growth mechanism of neuron, as I said, plasticity, actin and microtubules plays a very important role. Okay, here this neuron is growing. And interesting thing is that single cell organism like paramecium swim about, avoid obstacles and predators, find food and mate, and have sex, all without any synaptic connection. So these are single cellular organisms. They don't have any neurons, so no synaptic connection. And they use cytoskeletal structures such as microtubule in protruding cilia and within their internal cytoplasm for sensing the movement. So once they sense, the cytoskeleton decides how to move to push the cell wall. And here I show unicellular stentor rosali. They feeds on bacteria, algae, some flagellates and other ciliates if disturbed. And now you see here somebody is disturbing with some polystyrene bead. It gets disturbed, and once it gets disturbed, it detaches from there and run away. So it makes mind to detach and swim away from the site. So see this sort of intelligence, like it is responding without any neurons, okay, and without any synapses. And this is the schematic structure of this. So it is believed that this unicellular organism came together and form a collection and form a multicellular organism, and some portion of this multicellular organism, some cells have some specialized function, and some portion of the cell, some cells, they become photoreceptor, photosensitive, and then slowly through evolution it has formed a complex structure and more complicated eye structure. This is a very small microorganism and it has two eyes here. So this complexity or this evolution happened for better survival. I will not go further beyond this. So by now you might have got some idea how from unicellular organism you got, due to collection of this unicellular organism, you got a multicellular organism, and different portion of that organism were specialized for different function. Okay. So here I have shown this bacteria culture, and it has different antibiotic concentration. So very high concentration is in the center, and there are different bands of different concentration, and you see how bacteria evolves and can get used to low concentration of antibiotic. Then again some mutation, higher concentration, it will survive on the higher concentration. So they then finally become antibiotic resistance. So here I just show quickly the evolution of multicellular organism, what I was saying. And this is amoeba. Okay, you have Eugela, paramecium, then Hydra. Okay, more complicated or complex organism started evolving. And so this, like in the sea, mostly the living thing, it is believed that started in the sea and then they came to land. So now these are tortoise, dinosaurs, a bird, okay, and so forth. There are mammals have come later on, and now you have other animals, and then of course finally you get man. Okay. So thank you, and we shall see you again in part 3 of 3, which will deal with brain, neurons, and consciousness.